Welcome, everyone. I'm Jim Carrington, President and CEO of the Danforth Plant Science Center, and I thank you for coming to the 4th edition of Big Ideas. And I hope I'm not spoiling anything, but I think you're in for a real treat this evening. 9 young Danforth Center scientists working in 3 teams have been honing 3 big ideas. Ideas that, if developed with sufficient resources, with enough time, and if implemented, would impact hundreds of thousands or millions of people. Ideas that address real challenges in the environment and real challenges with the food that we need. Ideas that would truly deliver on our mission to improve the human condition through plant science. The teams built ideas based on work that they're already doing in their groups, in their labs. But they were asked to expand upon their work with a vision about where that work could lead. The teams are composed of graduate students, research assistants, postdoctoral fellows, and statisticians, all early career. They're not the ones you're used to hearing from. The teams built ideas, each team committed to convey their big idea to you, an audience that lacks their detailed scientific knowledge, that lacks their complicated vocabulary, and lacks their motivation. Their challenge was to communicate their big idea to you in an engaging and inspiring way. We're honored that you've joined us for tonight to experience the very best that the Danforth Center has to offer. So, to get this show rolling, let me introduce and welcome to the stage the Danforth Center Director of Phenotyping and Principal Investigator, Katie Murphy.
Thank you, Jim. Good evening. Welcome to Big Ideas. Thank you all for being here tonight, and thank you to Genective for sponsoring the reception. You are going to meet some incredible scientists tonight. They are going to share their work with you and the potential it has to make all of our lives better. By the way, they agreed to make this a competition, okay? You, the audience, are going to select the winner. This is not just for bragging rights. There is a $10,000 research grant that is going to the winning team to further their big idea. All right, thank you. Yes? You are going to vote for the team presentation that you find to be the most creative, inspiring, and engaging. Here's how we're going to run the show. You will hear from each of our 3 teams, followed by questions from our panel. That's going to help you decide how to vote. Before we get started, I need your help. We need to test our voting system. We want to make sure this is working correctly. If you're here in the room, take out your clicker, but don't press anything. Okay? Not yet. You only get 1 shot at this, so please don't press anything. If you are joining us online, I got you. We already tested your voting system. You will get a link at the end of the show. Okay? So for those of you here in the room, listen carefully. I know whose clicker's whose. Okay? If this is your first time to the Danforth Center, we are so happy you're here. You are going to vote number 1. If this is your second time here, welcome back. You will vote number 2. And if this is your 3rd time here, we love you. Thank you for coming again. You will vote number 3. Ready? Vote now. All right, while the system is calculating, I just want to tell you that as a scientist, I love this event. Okay? I feel so inspired every day when I come to work and I see my colleagues and my friends use plant science to make the world a better place. I'm so excited that tonight you are going to hear their ideas and their passions brought to life on the stage. Because I know if you are even a fraction as inspired as I am, you're going to leave the night feeling hopeful for the future. All right, I'm looking for my signal that our clickers are working. All right, we got thumbs up. Thank you very much. Okay, are you ready to meet our illustrious panel? Yes, thank you very much. First up, we have the managing partner of Edward Jones and the chair of the Danforth Center Board of Directors, Penny Pennington. Next, we have the founding partner of Lewis and Clark Ventures, an agribusiness investment firm, Tom Hillman. Last but certainly not least, we have the former president and CEO of the Carnival Corporation, Arnold Donald. Okay, panelists, buckle up. Are you all ready for big ideas? Let's hit it.
Plants feed the world, and the sun feeds the plants.
Plants capture only 1% of the total sunlight that hits the earth.
Can we build more productive crops that use light more effectively? Our big idea is plants that grow in the dark.
I'm Selma Adam from Jersey City, New Jersey.
And I'm Marcus Griffiths from Birmingham, England.
And I'm Vanessa Jawa here from Hillsborough, Missouri. And we are Team Sun Solutions from the Newsonout and Top Labs.
We have a technical glitch. We are looking for the clicker.
Oh,
It's like my couch at home.
wow. Wow.
I have a confession to make. I've killed before. And I'm going to do it again.
Selma, come on. Plants only need 2 things to live, water and light. But I get it. Keeping plants happy is harder than it seems. I can control how much water I give my plant, but controlling light is much more difficult. HGTV lied to me. They made me think I could decorate my whole house with plants. Instead, they're all crammed into my single south-facing window. If I move them away, they get tall and spindly, and they bend towards the window because they can tell they're getting less light. The plant is doing this for a good reason. Its goal is to survive, so it's growing this way in an attempt to get closer to the sun. A young tree in the forest grows really fast because it's completely shaded by the taller trees, and it needs to get to that sunlight. If it doesn't survive. So this elongation is actually a fight for survival. But my houseplants, they're not in a forest, and I know they're getting enough light to live, but they grow as if they aren't.
And this problem doesn't stop at houseplants. Light is one of the biggest limitations in where and how we grow our food. Have you ever wondered why we don't grow more food in cities, close to where people live? It comes down to land and light. But let's say we find some spare space to grow plants. The issue is that buildings shade those plants. It can be hard to get more than 5-6 hours of sunlight in your backyard, and never mind on an apartment balcony.
Now, light availability is a problem even in big open farm fields. Imagine you're on a freshly sown field, you see a tiny seedling sprout up. It's basking in the unobstructed sun. Then another sprout emerges, and another. Our tiny seedling is no longer an only child. And just like siblings, they begin to squabble over the sun. But a plant can't get up and move to a better location. All it can do is grow. So this plant is going to start trying to outgrow each other, trying to get to the sun. The taller they get, the more they block the light from each other. And in doing so, they're wasting their energy and resources on competitive growth instead of yield. This fast kind of growth was good for that sapling tree in the forest. Stretching to survive made sense. But it's bad for our sibling sprouts on the farm. It reduces yield. What's good in the forest is bad in the field. Now, light is really important. And I don't think I can control the sun, but I might be able to control plants. So what if we make a plant that doesn't care about how much light it gets? What if we make a plant that can grow in the dark?
Oh my God, Vanessa, you've lost it. Growing a plant in pure darkness would be an incredible achievement. But what we mean when we say grow in the dark is more like grow in less light. So imagine a tomato plant. It needs 8 to 10 hours of sun every single day. But what if it didn't? What if it just needed 4? If we can create a plant that thrives in low light, we'd be able to overcome the major problem of growing food, which is critical given our expanding world population. In the US, 40 to 50% of all fresh produce spoils in the transition from farm to consumer, mainly due to issues in the supply chain, like storage, transportation, and distribution. One option to make up for these losses is to increase farmland, but that means cutting down trees, clearing savannas, and just destroying entire ecosystems. Almost half of all habitable land on Earth is already used for agriculture, and we don't want to keep expanding that.
So if expanding farmland isn't a good idea, can't we try planting more crops in the same space that we already have? It works to a certain extent, but ultimately we come back to the same problem again, which is light and light availability. Packing more plants into the same space does not necessarily mean more food, and that's because when plants are crowded out, they start competing for resources, for water, for nutrients, and especially for light. If we look at a single plant that was grown in high density, this plant would have fewer branches, looks weaker, and ultimately yields less. This is because that plant's priority has become competitive growth at the expense of being productive.
Indoor vertical farming could be a good option. It has the benefit of bringing large-scale food production close to where people live without expanding farmland, but it has a really large cost in terms of energy consumption. These farms are estimated to consume more than 7 times that of greenhouses, and over 55% of that is attributed to light usage, even if they're using LEDs.
We are going to make a plant that is strong and healthy, whether it's in a field, in an urban garden, or in Vanessa's living room. Our plant won't stretch to reach for the sun, so by preventing this shape-shifting, we'll be able to keep plants compact so they don't shade each other. Our idea will prevent unnecessary competitive growth and instead promote yield and productivity. So basically, these plants won't overreact when they're on a light diet.
The good news is we know a lot about how plants sense light and why they grow the way they do. Plants have a light-sensing gene called phytochrome B, and this gene basically acts like the plant's eyes. It turns on in the day and turns off at night, and that's how the plant keeps track of the time. And we can also change this gene so that it tricks the plant into thinking it's daytime, even when it's not. And then we're also pursuing on 2 other genes, one called PIFs, that controls how shoots elongate, and another gene called PINs, that controls how the shoots bend towards the light.
Since we know the genes, we can tweak them to function differently in a way that's going to help the plant. We are going to use a revolutionary tool called CRISPR to edit these target genes. CRISPR allows us to edit DNA much like we can a Word document. We can easily delete a word or change a letter, allowing us to precisely control what that DNA does. For example, let's take this sentence, "Let's eat, Grandma." If we remove a single character, we change our dinner invitation into a Hannibal Lecter situation. Let's eat, Grandma. Small changes to the DNA can have a big impact. We plan on using CRISPR to fine-tune our genes like a dimmer switch to turn them on, off, or something in between.
Evolution and breeding are based on genetic changes just like this. But breeding takes many years and can change many genes at once, so it takes a long time to get the right set of genes into a single plant. Humans have been breeding corn for over 10,000 years, and we're still trying to perfect it. We know exactly which genes we want to target, we know how to do it, and we can be out in the field within the next year or 2. And surprise, we've already started. We've modified the vision gene, phytochrome B, to stay in the always-on position, meaning the plant thinks it's always in the sun. So even when growing in low light, the plant stays compact and healthy, as if it was grown in normal light.
We've even gone on to edit our second target gene that controls elongation. So in low light, this plant remains compact, allowing us to plant it at higher densities or in between rows of taller crops. Our results are so promising that we've even filed a patent on this. Our third target gene is the bending gene. And our edit will make it so that this plant doesn't bend towards the light. It stays upright. CRISPR allows us to combine all of these edits into a single plant.
And we're not stopping there with the leaves. The roots of the plant also matter too. Did you know that below ground, the roots of the plant can also sense light? In low light conditions, the plants prioritize their shoot growth at the expense of the roots. And that's an issue because roots are what takes up water and nutrients needed for the plant to grow. For a healthy plant, we would want the roots and shoots to grow in balance. The light signaling genes that we're targeting, though, we're also finding that we can promote root growth as well with these light signaling edits. We're using image analysis to precisely analyze how our plants grow. We're measuring things like root length and root thickness, as well as leaf area and plant shape, all in respect to low light.
We are currently converting the cover crop pennycress into a bioenergy fuel source. This crop produces oil that's then used to make sustainable jet fuel. And the best part is it grows over the winter, so it doesn't compete with other food crops. But pennycress needs to be planted in September, and fields around the US can vary greatly in how they look at this time. Pennycress needs to establish itself in the soil amidst rows of corn, which can reach up to 9 feet tall. So our work began with trying to help this crop overcome the real-world light challenges that it faces. Pennycress is a member of the cabbage family, along with canola, kale, and broccoli. So our gene strategy can be quickly applied to these closely related species, and later on adapted to more distant crop species like wheat or rice.
Our 3 targets allow us flexibility and precision. We are building a genetic control panel where we can turn our genes on, off, or dim them in various configurations. Different environments are going to call for different panel settings. Pennycress growing in standing corn, we might want to edit multiple genes. But pennycress growing in corn stubble, maybe we want to keep that bending gene on, allowing the seedlings to bend around the corn stubble for establishment. Our strategy is going to vary based on the species too. Cabbage, we don't need to worry about bending. We still need to control elongation. Our targets allow us to fine-tune our strategy to accommodate both the plant and the situational needs.
So our big idea, growing plants in the dark, would help solve a big agricultural challenge. Our genetic control panel will allow us to grow more food more productively and sustainably using the same amount of land, just by being able to plant at higher densities. And also allow us to grow food in places that used to be off limits, such as in the inner city. This means fresher food, less spoilage, less trucks on the road, and ultimately cheaper food for everyone. And then with indoor farming on the rise too, our edited plants would need less supplemental lighting and would therefore have a lower energy cost for growing food.
Now, this sounds like a great idea. It's important. It's reasonable. This isn't reasonable ideas. This is big ideas. So how about plants in space? On a spaceship? Yeah. On a spaceship, no natural light. On other planets, different amounts of light. If we're ever going to colonize Mars, it's important to consider that Mars only receives 44% of the light that Earth does. We believe our glow-in-the-dark plants can remove even the terrestrial limitations on where we can grow plants. Now, this is all a big idea, but it's not just a pipe dream. It's already working. We would like to continue to pursue our gene targets to make a plant that can grow in the dark. By doing this, we're solving a global agriculture problem and an energy problem.
A sustainability problem and a food security problem.
A houseplant problem, and most importantly, we'll be ending my killing streak.
Great job, y'all. You took us on an emotional roller coaster ride. First, I felt bad for houseplants. Then I felt bad for Grandma. And now I'm super excited about going to space. Okay, so you laid out a great big idea. Where is the first big application? It's probably not space. Where are you going to start, and with whom to apply this idea?
I think the first real world use is being able to plant more in like our fields. And being able to plant at higher density is the easiest way to actually produce more. We already have the infrastructure to do that and the land. We don't need to make anything new for that. We just need to get more seeds in the soil to perform better. And so we're actually trying to transition our seed with pennycress into to work with the company Covercress to actually start testing this. So we're developing partnerships to do these field trials.
I agree with Penny. It's a big idea. It's solving a lot of problems on our planet. Do you see that you're going to have plants or row crops or different areas where it's going to grow faster? It's going to be the same timing. I'm just curious what you think of the outcomes other than being able to grow something with less light and more densely.
I think with pennycress specifically, the US Midwest is like the initial area where pennycress would be grown. That's where it would grow very well. But we're changing the light signaling genes as well. We should be able to expand the area in which you can grow this plant and crop. But the initial would be in the US Midwest.
So that was not just a big idea, it was a big presentation. You guys did it very well. That was outstanding. There are other nutrients involved. So if you increase density, there's other competition going on besides for light. And so the question becomes yields. So as you change these genes, what are you actually doing to what's going to be yielded, even if you have increased density? Have you been able to test any of them?
You're talking about being able to better use nutrients?
Yeah, in other words, if you have greater density, it's more plants competing for fertilizer.
Yeah,
and,
that's why we have our root expert.
Right. So what happens? What about yields in the
I
end?
think
Have you really improved yields in the end? Or have you just changed the light profile?
I think it's like dual purpose, really. One could be high impact, high input agriculture, so want to get more yield, and that's where bigger plants, growing high density plants. But also the other application would be low density or low input systems, where maybe you want to change that genetic control panel, pick different genes or different levels of genes to try and optimize it for that particular agricultural environment.
Excellent, thanks.
We really hope it'll end up with higher yields. But we have to test that. So we're working on that part. Good
job, y'all. Thank you, Team Sun Solutions.
There are 1,300 sites across the U.S. where pollution has forced people to abandon their homes and communities.
Cleaning up these sites can cost billions of dollars and take decades.
Could plant science be the key to solving this massive problem?
My name is Josh Sumner from St. Louis, Missouri.
I'm Anna Casto from Golden, Colorado.
And I'm Keely Brown from Spokane, Washington. And we are Team Bioaccumulate from the Guillen and Fahlgren Labs. Who has heard of the garbage island, 3 times the size of France, floating somewhere out in the ocean? And I'm sure you've all seen the dish soap commercials with the sad birds covered in oil. These large-scale pollution events make for dramatic visuals, and the resulting ecological destruction is emotionally visceral and very important. But it can sometimes feel kind of far away and like someone else's problem, But here in Missouri, even right near St. Louis, we have plenty of our own pollution to deal with.
Anyone else here grow up in eastern Missouri? Just 30 minutes from here, in 2001, dust off the street in Herculaneum was tested by the state and found to be over 30% lead. Later that year, the EPA found elevated concentrations of lead in the air as well. Subsequently, over half of young children in the affected area had high levels of lead in their blood, which can have dramatic health consequences, such as learning impairment, developmental delays, and mood disorders. Eventually, the Doe Run Company, whose smelting facility was the main source of lead pollution, had to physically remove contaminated soil from over 750 parks and homes as part of a massive cleanup effort. But Herculaneum is just one example. There are dozens of toxic sites called Superfund sites by the EPA, flagged for hazardous waste disposal across Missouri. And across the US as a whole, there's over 1,300 such sites.
Wait, are you saying that they just picked up the dirt and moved it somewhere else? That sounds hard and expensive.
Yeah, they spent almost $300 million on cleanup. Heavy metals like lead, they don't just go away. They'll stay in the soil for a very long time. The best way we've come up with to fix lead-contaminated soil is to dig it up and move it to a safer disposal site.
And that's the problem we want to solve. We want to use the power of plants to remove contaminants from unusable soil and decrease the consequences of land contamination. Plants are, thank you. Plants are already great at absorbing things from the soil. They take up the nutrients they need, which is why your lawn does better when you fertilize it. But they can also take up pollutants, and some are even resilient enough not to let those harmful things affect their growth. Wildflowers in California are known to take up copper from the soil around abandoned mines, and ferns and mustards have already been used to clean up arsenic and lead from the soil around the Barber Orchard Superfund site in North Carolina. But all of these plants have had to adapt to the reality of growing in less than ideal conditions. They haven't been specifically bred to be good at it.
This is why our big idea is to make plants better at removing dangerous toxins like lead from the soil. But that might be a little bit too broad, so we want to start with a specific plant.
You both know I'm a statistician, not a plant scientist. How would we pick just one plant to start with?
Well, we need something that's going to be easy to harvest and remove from an area once it's done cleaning up the soil. We need something that's not going to become invasive and compete with plants that are already there. And we need something with a lot of varieties available already so we can pick the ones that might be best. Crop plants fit a lot of these requirements, and in fact, quinoa, a grain crop, is already grown on soil that's too contaminated with salt for anything else to grow.
You might be familiar with quinoa from that one time you ordered some kind of healthy-looking protein bowl from St. Louis Bread Co. But quinoa is actually an ancient grain. It's popular in the United States as a protein-rich source of vitamins, and in countries like Peru and Bolivia, it's a staple crop. The plant quinoa is already pretty good at removing contaminants like excess salt, lead, and cadmium from the soil, but it could be much better. Imagine specific varieties of quinoa bred to clean up specific contaminants, just like we grow different types of grapes for different purposes. This is how we get different types of wine, red versus white grapes, grapes that taste like cotton candy, and seedless grapes. All of these varieties are the result of selective breeding.
Hold on, all those grapes come from selective breeding and not some fancier gene editing?
No.
See, I thought that selective breeding was something we used to make cool new kinds of dogs back in the day.
Yeah, exactly. Thousands of years ago, humans began by identifying traits in wolves that made them suitable for living with humans, like a gentle demeanor. By breeding dogs with good combinations of traits over time, we've arrived at this perfectly adapted specimen. Her ancestors were specifically selected for their increased support of my favorite soccer team. But it works the same way for plants. We want to breed quinoa that gets better over time at absorbing contaminants from the soil. But in order to do that, we're going to have to measure the amount of something like a heavy metal that winds up in the quinoa's tissue. And it turns out that the standard way of doing that is tedious and expensive. And it involves bringing plants from the field back to the laboratory, setting them on fire, irradiating the vapor, and then analyzing the qualities of those vaporized atoms.
What if there was a better way to measure the amount of lead in quinoa plants more quickly, safely, and affordably? In our lab, we use cameras to measure all kinds of physical characteristics of plants. But could a camera really show us how much lead is in a plant? After all, you wouldn't be able to tell just by looking at it, right?
Who here has ever used a black light? In a dark room, things look different under a black light compared to the same well-lit room, right? Or maybe you've used a thermal camera in your house and been able to see the hot water pipes in your walls. Or, admittedly, if your drywall work is as shoddy as mine, you just see cold air pouring in through all the cracks. Or maybe during a nature documentary, you've seen animals running around at night under night vision.
Thermal and infrared cameras use different parts of the same light spectrum to help us see things that we can't normally see. Humans can see only a small portion of this spectrum. So using specialized cameras and lenses, we can reveal things that are only visible outside of the range that humans can see.
For example, if I put on infrared goggles, I could tell who here has washed their clothes most recently because laundry detergent has a distinctive color in the infrared range. Alternatively, if we turned on a UV light, we could see who's washed their hands most recently because soap residue will shine in the ultraviolet range. There's so much more information in light than just what we normally can see. To make use of that, we'll employ a specialized camera called a hyperspectral camera that can see way beyond what humans can and that will tell us which quinoa plants are doing the best job of pulling up environmental contaminants like lead.
In this way, we can more quickly, safely, and affordably pick the best types of quinoa. We can even take these pictures in the field without having to bring the plants back to the lab, which will help us accelerate the process of improving quinoa's natural abilities.
Now that we've decided to start with quinoa, and we have a way to measure the amount of pollutants in the quinoa plants, how do we actually get to a variety of quinoa that's optimized to clean up the soil? As we mentioned, crop plants are a great starting point for breeding because a pool of genetic diversity is already available. I could go online today and order seeds from hundreds of varieties of quinoa from all over the world. We can plant those seeds on lead-contaminated soil, measure the plants with our hyperspectral camera, and then select the plants that take up the most lead with the least effect on their growth. We can take those best varieties, cross them together to combine their effects, grow the new seeds out again, and then rinse and repeat over many generations. So at this point, you might be thinking, didn't Keely just say that it took thousands of years to breed dogs? There are so many contaminated sites that need to be cleaned up as soon as possible. We can't wait decades for the perfect variety of quinoa.
This is somewhere that biostatistics can help. Sparing the gory mathematical details, these are types of problems that statisticians have gotten very good at solving over recent years. One of the reasons why selective breeding experiments can be slow is that we're not always sure what the outcome of a cross is going to be. But what if we could be a lot more sure that in each generation, we're picking the best plants to cross together to get towards our eventual goal of an ideal plant for removing contaminants from the soil? Well, in fact, we can, because we can collect and use a ton of data very inexpensively.
You've probably seen Netflix suggest new shows to you based on shows you've already watched. Netflix is making predictions about what you'll like based on all the data that they've collected about each show, about you, and about people like you. This way, you can find new content quickly without spending hours scrolling through thousands of options.
With recent advances in DNA sequencing, as well as our image-based measurements of quinoa plants, we also can collect a lot of data very quickly and very cheaply. That data will let us make predictions about the outcomes of crosses without actually having to perform all of them. And for those that we do perform, our image-based measurements mean that we don't have to wait around a long time to see how well that plant does. Through clever application of statistics, cutting-edge imaging, and AI, we can take advantage of a ton of data to quickly select the best plants in each generation and could start testing promising varieties of quinoa later this year.
Besides speed, another major benefit of this breeding strategy is that we don't need to know which genes are involved in making quinoa better at taking up contaminants from the soil, which is great news because it took decades to find the just 6 genes involved in corn domestication. But we don't have to understand any of the complicated mechanisms for why quinoa might be improving, only that it is getting better over time at what we want it to do. And this process can happen a lot faster than you might think. In just 1 year of selective breeding, corn yields typically increase by 2%, which doesn't sound like a lot, but in the US alone, that amounts to $870 million extra dollars every year.
I have just one more question. If our plants are taking lead out of the soil and into their leaves, then what happens to the plants? Aren't we back to having to cart a bunch of debris off to some disposal site?
Sure, but plants are a lot lighter and more compressible than 750 lawns worth of soil like in Herculaneum. It also prevents the neighborhood park from being destroyed when all that soil is carted off. Like we mentioned, plants are already great at absorbing things from the soil, even when they're present in just small amounts. This way, plants can concentrate contaminants away from the soil, and then the plants and contaminants can be easily removed by harvesting the plants. A big advantage to our approach is that we can plug into disposal processes that already exist for remediation, but we can make it much less costly and much more environmentally friendly.
Pollution is a big problem with human health consequences and very expensive solutions. We want to use the power of plants to clean up the environment and make it safer for us all. Our big idea, to breed quinoa that is better at taking up things like salt and heavy metal from the soil, will help people where they are, impact human health and agriculture, and most importantly, is readily achievable with the tools and expertise in our labs.
This is really a fantastic idea. And when I think of cleaning our planet and the environment through plants, it's just very noble and worthy. I'm kind of a merger and acquisitions person, and I already see an opportunity to use sun solutions in combination with what you're doing. So with less light, you could grow it on any field.
Yeah.
But seriously, just share with us your story. How did you end up thinking of this idea to come up with?
I can start with that as the non-plant scientist. But growing up in eastern Missouri, there have been tons of different stories of just the environment getting destroyed, be it Herculaneum or like the area around Pruitt-Igoe, all these sort of nasty situations. And then when I was in, I think I was graduating high school, my family got a small farm, and the amount of work to fix just destroyed soil, be it because it's been depleted from poor farming practices or there's lead and salts and other nasty stuff in it, was dramatic to see.
In our lab, we actually work with quinoa already, and so quinoa, we knew, has these specialized small bladders on their leaves to help take up and store things like excess salt so that it doesn't affect the quinoa's growth as much. And so we thought we could harness this sort of natural ability of quinoa already and perhaps apply it to new things to clean up like lead or cadmium.
Outstanding big idea and really outstanding presentation. So how long? So if you had funding and you had everything, how long would it take to clean up a super fund? So you say you want to accelerate the time. Let's say it all worked.
Yeah, I think it could be really quick. I think the timeline from ordering the seeds, like Anna said, we could do that today, to measuring them all. It would just be the lifespan of a plant, figuring out which genotypes might be best. It's possible that there are some that grow in different parts of the world with different environments that are already better than the ones that we grow for food here at cleaning up things like lead. We could get those in the ground as soon as we know which ones are best. So like Josh said, later this year in places that need it. And I think the important part of this idea is that it doesn't actually, we want to get to the perfect variety of quinoa, but it doesn't take perfection to make an impact. We could put the best genotype that exists in a field next week, and it would make a difference.
And from the statistics side, how many years of growing would it take to clean up a site,
Well, we aren't totally... I'll speak as the non-plant scientist and give them time, so I'll step up here. We are not totally sure, but that will depend a lot on the exact Superfund site. So last summer, it seemed like every week a train was tipping over with toxic waste. One of the things that, as Keely's alluding to, that's really cool about this is we can parallelize all over the country. Quinoa can grow in all sorts of conditions. So we could put it in different places and just keep iterating very quickly. Some places will need to grow out deeper roots. Some places will have very superficial contamination and could happen much faster, say in a growing season. And now I'll step back and let them go.
Math guy, thanks for explaining why you're there. That really helps. So
I know it wasn't obvious before.
I did have this question, and y'all kind of answered it. You used to have poisonous soil, now we got poisonous plants. What do you do with the poisonous plants? You kind of answered that. You said we're going to use existing remediation to deal with that. You also got to have somebody who's going to plant that plant. So I'm thinking about all of the after ancillary effects of poisoning other people, other places. You've got a NIMBY effect, not in my backyard. Don't put an incinerator in my backyard. That's getting rid of these plants. So, how do you see the extensions that need to be made for this to actually work?
I can start with this one. So we, I think one important thing is that we expect sort of our customers of the perfect quinoa variety to be companies like the Doe Run Company or the government, places like the EPA, who are already responsible for doing this cleanup. And so as Anna mentioned, the cleanup already has to happen. And so if their maintenance practices have to transition from dirt, which is easy to aerosolize, it's easy to breathe in as it's being transported, to something like plant material, which is a lot more compact, it's a lot more easy to contain, then I think those on the people whose responsibility it is to clean up, I think those management practices can be built into what happens after the plants grow.
Also, with respect to the not in my backyard thing, I suspect that growing some plants around would be much more attractive than having bulldozers coming by and digging up the soil. So I think combined with this potentially being a more cost-effective way to clean up the soil, it could also remediate some or prevent some of that not in my backyard attitude.
And with the current methods, landfills just keep filling up, but here we would have more like a permanent composting situation happening. Well, hopefully not permanent, hopefully we stop needing it eventually.
Thank you so much.
Worldwide, 30% of all crop production is lost due to plant diseases.
Plant diseases threaten farms, our livelihoods, and our food security.
Is there a better way to protect and sustain the crops we rely on? Our big idea is to harness a plant's natural defense mechanisms to protect crops from disease.
I am Anastasia Klebanovich from Kamenets-Podilsky, Ukraine.
I am Ali Ingermeyer from Darmstadt, Indiana.
And I'm Sam Newsy from Pompano Beach, Florida. And we are Team Poor Decisions from the Zimic and Birch Smith Labs. Many things can be said about Florida. It's the state where I was born, but it's also the state where a man once used an alligator to rob a Wendy's. While these Florida man stories continuously build a certain reputation for Florida on the internet, Florida is known for something far more valuable. It's citrus fruits. Growing up there, I remember summer days spent in the backyard of my grandparents' house, eating fresh oranges straight from their tree. I was too small to reach the oranges at the top, and my grandfather would lift me up so that I could pick them, myself, being the independent child that I was. I also remember the day the Florida Department of Agriculture came and cut the tree down. They told us they were cutting down the tree to contain an invisible threat.
So, what is happening to our oranges? Plants get sick, just like we do. But while we can go to urgent care to get medicine, a disease in plants is almost always a death sentence. Over the past decade, the citrus trees in Florida have been ravaged by microscopic enemies, viruses, bacteria, and fungi. Infected citrus trees produce fewer and smaller fruits until they slowly wither away and die. And in a field of hundreds of citrus trees packed together, an infection spreads like wildfire. By the time we can see disease symptoms with the naked eye, the tree is not going to survive. The visibly infected tree and all of the surrounding trees must be cut down to prevent the disease from spreading. If we take a closer look at citrus production rates over the past decade, Florida alone produced 70% of the oranges consumed in the United States. That's 200 million boxes of oranges. To put that into perspective, imagine filling Busch Stadium with oranges and then filling it again 6 more times. But last year, Florida's production rate dropped to only 10% of what it was just a decade ago. And we feel that in our wallets when we go to buy orange juice for our bottomless mimosas that actually are not so bottomless anymore. But this isn't just a Florida problem. It's happening worldwide.
Maybe you are not a big fan of oranges for breakfast. So why should you care? The citrus trees are not the only crops at risk. In the 50s, we almost lost the entire grow-mish-out banana variety. And now the modern Cavendish banana faces the same threat of extinction from a fungal infection. But plant diseases don't just threaten fruits. They are responsible for almost 40% of global losses in staple crops like potatoes, wheat, and rice. That loss adds up to a staggering $250 billion lost every year. Think about it this way. Every time you blink, it costs the world economy almost $7,000. So please, stop blinking. Well, this crisis is not just about the price of orange juice or the future of bananas. It's about food security and human lives. We face an extreme challenge. As our population grows, our increasing food demands cannot be satisfied if staple crops are lost to plant diseases.
Viruses cause many of the plant diseases we see in fields today. We wanted to step into the metaphorical shoes of a plant virus to better understand how plants defend themselves. We sat down to brainstorm, and after hours of thinking, we had nothing. So we decided to blow off some steam and go to an escape room. Now, some of you may be thinking, who goes to an escape room to relax? Just us, apparently. Could you please raise your hand if you've ever been to an escape room? Wow, okay, a lot of you have. Awesome. For those of you who haven't, here's how it works. You and your team are locked in a room filled with puzzles that you must quickly solve to find a key that unlocks the door before time runs out and the room explodes. Sounds fun, right? Well, let me tell you how it really went. The moment that door slammed shut and the timer started, it was pure chaos. There's a puzzle over here, and a lockbox over here, and oh my gosh, what are you doing? Are you even trying? Do you want us to lose? Our dear Sam, she did find a key, but it was actually the key to her car, not the escape room door. At only 15 minutes in, we were just desperately trying to escape the room, and each other, if you know what I mean. But that's when it hit us. This is how a plant virus must feel trapped inside of a plant cell.
Picture us desperately trying to escape that room. That's the virus searching for the next door. If we imagine the escape room as one plant cell, the doors are extremely tiny channels called pores. And these pores are not left wide open all the time. They are tightly regulated by molecular locks, different pore molecules controlling what comes in and out of the cell. In healthy plants, thousands of cells send important messages to each other through their pores. All pore decisions are carefully made so that the plant can grow and thrive. So what happens when the virus breaks in? It must hijack pore molecule, open a pore, and cross into the next cell. Let's zoom in to have a closer look. Inside the plant cell, virus does two things. It multiplies, and it spreads to other cells through their pores, repeating this process over and over to infect the entire plant. So when plants sense the virus, they try to lock their pores to contain the viruses inside. But that is quite challenging. Imagine locking hundreds of doors at the same time. So how can we help the plant successfully defend against the virus? What if we could build an impossible escape room? One where the virus could not open the doors, no matter how hard it tries. Our big idea is to fortify natural plant defense strategies to prevent the spread of plant viruses.
Some of you might wonder how we can see a disease before symptoms show up. It's not as easy as simply asking a plant if it doesn't feel well. Plants are terrible at small talk, so we have to run tests. In the lab, we use tomatoes for our experiments since they grow fast and are susceptible to disease. Think about what happens when we get sick. We go to the doctor. If the doctor needs to see more details, we might get an MRI or a PET scan. Giving a plant a PET scan may sound like a silly idea.
Or a poor decision.
But we need powerful imaging. Because first, we need to watch how the virus breaks in. A magnifying glass or even a tabletop microscope aren't good enough to see because the pores and the viruses are incredibly small. We're talking about objects that are 10,000 times thinner than a single human hair. But here at the Danforth Center, just down the hall, we have the technology to watch a virus inside of a plant cell. Our high-powered microscopes can magnify nearly 1 million times to reveal the inside of a very small pore and even individual viruses. We can catch viruses in the act of picking the block and breaking into the next cell, like molecular surveillance cameras. But watching viruses move from cell to cell isn't enough. We need to know what is letting them through the pore. How can we tell which pore molecules a virus uses to successfully break into the next cell? In tandem with our high-powered microscopes, we use a molecular technique called proximity labeling. It works just like it sounds. Anything close by gets labeled. Zooming into the pore, we can see its structure. First, we place a marker, shown here in blue. Then, in the presence of a virus, some molecules, in red and purple, help the virus move through the pore. The marker will label anything close by, shown here with the yellow tags. This approach allows us to successfully pinpoint all the nearby pore molecules that help the virus move from cell to cell.
So now that we know how the virus is breaking in, how does that help us?
So now we can work on applying what we've learned to protect plants.
Maybe we could jam up the pore lock so that even with the right key, the virus couldn't open it. We could stick a wad of freshly chewed gum into that lock. Then the virus wouldn't be able to open the door and leave the cell.
Not all ideas can be Nobel Prize worthy. Allie, tell them about small molecules instead.
Our innovation is a custom-designed molecule acting as a blocker to interact with the pore lock. We'll design this blocker to be stable and small enough to slip into the pore molecule. Our blocker will be able to fit into the pore molecule and prevent a virus from attaching to the pore and slipping through. What we give plants is a precision tool that allows them to prevent the virus from moving through their pores into the next cells, but doesn't stop or interfere with their day-to-day communication.
Our blocker is designed as a field or a greenhouse spray, a solution containing these protective mini proteins. It would be very easy to deliver our blocker to any infected plant, but also to the surrounding healthy plants.
Farmers could spray it on their crops like they would a routine treatment. This blocker could be applied to crops in the field and to garden plants. From feeding the world to guarding your backyard tomato patch, our technology adapts to the scale.
Imagine if my grandparents had this blocker. Perhaps the trees from my childhood would still be around today. With our big idea, we want to prevent and treat plant diseases. So let me ask you a question. If you had a choice, would you rather eat this tomato or this one? You'd pick the healthy one. Spoiler alert.
Yeah.
Making the right choice starts long before the harvest, through our big idea, and yes, a few poor decisions.
Sorry. I'm so sorry.
I messed up too.
All right, poor decisions. That was a great presentation. You guys know what a homophone is? Yeah, you do, right? Well, that was a clever use of a homophone.
Thank you.
In poor decisions. Thank you. But you did not make a poor decision in choosing poor decisions as your team. So in any event, this is a big area of research, and because it's a mega problem, as you articulated. So others are working on this, I'm sure. So how is your solution unique versus others that you might be aware of?
Go ahead. So, one of the great things about being at the Danforth Center is that there actually is a startup here currently that is working on fungicides to prevent fungus specifically. But ours is inherently different because of the mode of infection. So fungi and viruses, bacteria are all infecting plants differently, and ours specifically is going to target viruses, as opposed to other companies that are looking at other kinds of methods of action, essentially.
Yeah.
So method of action is
yes.
differentiated. Have you seen Last of Us?
Yes.
Stick with virus or stay away from the fungi.
Absolutely, absolutely.
Yeah, I mean, having citrus farms is just really vital for everyone. I'm trying to understand the application. Is this a blocker that's done at the planning level, or is it also involving with spraying? And if you are doing that, does it get included with other materials that people use to spray to combat other kinds of diseases?
So I think that would depend on which molecule is helping the virus move through the pore. So that depends on, so that is what hinges on how we design our blocker. So I think how, and then from there, the composition of the blocker would then determine if you can include it with other things, if it could be sprayed alone, things like that.
If I could add, what stands out in our research is that it's highly tunable system. So it can be customized based on the virus, but also the crop. And as we mentioned before, it should not interfere with regular or routine treatments, maybe, I don't know, other chemicals that farmers decide to spray on their crops, or the fertilization of the soil.
Sam, thank you for explaining to us why this is so near and dear to your heart. And I love the picture of you when you were little. So inquiring minds here want to know, do you really have an orange tattoo on your arm? I mean, you talk about riding for the brand.
I do, actually. And I actually did joke with a lot of people that I got the tattoo for this event. I did not, for the record, but I did play that joke on a few people.
Well, you just played on the psychology of the people who are about to vote. I mean, she took a needle for this big idea.
Thank you, guys. Thank you, guys.
All right, let's hear it one more time for all three teams. They can hear you cheering. Okay, it is that time of the night. I want to remind you how you're going to make this very challenging decision. Please choose the team presentation that you found to be the most creative, inspiring, and engaging. If you are joining us online, we're about to show you a QR code on the screen or a website that you can go to. You can place your vote online. For those of you in the audience, pull out those clickers, please. If you would like to select Team Sun Solutions, you're going to choose number 1. Team Bioaccumulate, number 2. And Team Poor Decisions, number 3. Please vote now. All right, while we tabulate these votes, I'd like to welcome Jim Carrington back to the stage.
Thank you, Katie. Is everybody having fun? Yeah. Now, because we have a few minutes to kill, I want to answer a question some of you might be wondering. What happens with all these ideas after the presentations? This is the 4th installment. We've done 9 other presentations since 2017. Well, just about every big idea leads to a new project or a new set of projects in these labs to either further the idea or something else related to the big idea gets proposed, proposals get submitted, grants are funded, and work expands in these areas. In some cases, the entire focus of the lab has shifted to those big ideas. There's one I like to call out specifically, and that's Keith Slotkin's lab. But there's much more. The winner of Big Ideas in 2023, Team Metablify from the Ivan Baxter lab, took their idea and created a new company, a new startup last year to commercialize their unique technology that was proposed in the big idea to better understand important metabolites. And can you guess the name of the company? Metablify. The winner from 2019 was a team from Keith Slotkin's lab. Just this week, 2 days ago, we announced a new startup company called Spearhead Bio, founded by Keith Slotkin. That company is developing and applying a completely unique way to do genome editing to create new traits in plants quicker and more precisely than existing technology. That company, Spearhead Bio, has its origins 6 years ago at Big Ideas. These ideas really do move forward. The ideas you heard tonight, if I had a bet, I'd bet they're going to turn into something big. And I'm really happy that you have all been here to witness for the first time those big ideas.
This is a TED Talk sort of format, and there's a big red circle up here, just like a TED Talk. Jim, I want to thank you for your leadership. I want to take just a moment to do this. The Danforth Center itself is a big idea, isn't it? Under Jim's leadership for a decade and a half, this big idea has been firmly planted and is flowering and flourishing. And Jim's leadership and his own ideas have established Danforth the world over to be the center of great ideas, mission-focused to change the world, impact the world positively through plant science. The Big Ideas initiative was Jim's idea. We're in the 4th installment of the Big Ideas, and the panel and I got to meet all these young scientists back in the back. We didn't ask them anything. There was no collusion going on about the questions that we were asking, but we asked them things like, why do you enjoy being at the Danforth Center? And they explained the impact and the people. And they also explained that they're some of the best in the world. They didn't quite say that. What they said was, we have other options for where to be. And in fact, some of us have been other places, have left the Danforth Center and came back. They are here because of the impact, because of the people, because of the leadership and the support that they're provided. They also told us to get ready for this. They've been working on it for 10 months. And they've spent 10 weeks. And they have spent significant time with each other, but also with Jim, which is the mark of great leadership. To be at the macro level of this enterprise, leading the way, but also to care about every individual scientist who's at work making a difference with their career. Will you help me thank Jim for his leadership?
Yeah. Thanks, Penny. Got to be loving that. Okay, that was way more than I thought. Thanks so much. Thank you. Well, that was very kind, and I appreciate those comments, Penny. I think part of your applause is because of what you've experienced tonight. You're already in a good mood. So I want to have the teams come back, and I assume since they're standing there, the votes have been tabulated and we have a winner. So come on up on stage. And I'm going to turn this back to Katie.
Okay. You just heard about the 2023 winner, Team Metablify. Let's welcome them to the stage. They have the great honor of announcing our winner. Team Metablify. Fortunately, they're very well practiced at this point. So, the moment we've been waiting for.
All right, well, it's tough to give up the crown, but if it's got to be done, what a group to give it to. And I accidentally took my clicker home from last year, so remember to give that back.
And now, without further ado, we'd like to announce the Big Ideas 2025 winners.
Sun Solutions!
Thank you all for coming to Big Ideas, and have a wonderful night.